Welcome everyone, my name is Edwin Gerald. I'm currently an undergraduate student researcher at the Inter-American University of Puerto Rico Aguadilla Precinct, and today I will be discussing with you a very important research that we're actually doing at BioPortal Solutions in Puerto Rico, and it's also in collaboration with the Cell Biology Education Consortium, so thank you so much. And this research was actually done through the principal investigators known as Dr. Elizabeth Padilla Crespo and Dr. Nathan Reyna, so thank you so much. So without further ado, let's start. The title of our research is called Using cBioPortal for Cancer Genomics for Research Investigation on Skin Cutaneous Melanoma. So it sounds very interesting. The main objectives of this presentation is for me to tell you how we were able to formulate a hypothesis using the cBioPortal for Cancer Genomics, and how we were able to actually come up with a conclusion on our research. This is the abstract of the official research. It's titled Using cBioPortal to Detect Genes Involved in Skin Melanoma, and a brief summary of the abstract is that we were able to identify 4 different genes, and these genes, I'm going to be discussing them later. And what happened is that using the cBioPortal for Cancer Genomics database, we identified the alteration frequencies of these genes in specific tumor samples. And each sample presented a different form of mutation, and if we summarize and we put all of these mutations into one, we were able to identify a complete roundabout approach to how we can actually interpret this genomic data. We concluded that the altered versions of CDKN2A, PCLO, LRP1B, and MGM, which are the genes that we selected, actually cause skin cutaneous melanoma, or they actually interact with one another to cause skin cutaneous melanoma. So it's important, before we actually continue with the presentation, to explain what exactly is skin cutaneous melanoma. Well, first off, it's a form of cancer. And just by skin or cutaneous, you know that it's actually located on the skin. It's important to know that this is a genetic disease, and it can be inherited. And by inherited, it means that it can go from generation to generation, from offspring to offspring, and so on. Because it is located on the skin, it means that it is formed by melanocytes. And melanocytes are the cells that actually compose the skin, and just by looking at the name, you know that they actually form melanin, which is actually the compound that is on our skin that actually causes it to be the color that it is. So what happens is that these melanocytes, when they are in an oncogenic state, these cells actually have oncogenic potential, they actually start to proliferate. And by proliferate, mean that they actually start to multiply uncontrollably, and then they start to form the tumor, which is actually this accommodation, this entirety of tumor cells that actually grow together to cause cancer. Here we can actually look at some pictures that were taken that actually are cutaneous melanoma. And if you can look at the particular color, you can see that they're actually darkened in color. And why is that? Why is it darkened in color? Well, what happens is that when you have a melanocyte cell proliferating uncontrollably on the skin, they start to produce more melanin, which thus causes this dark pigmentation on the skin. It's important to note that the diagnosis for this cancer is usually average of 65 years old. Usually 65 elderly people actually present this form of cancer. Now, this is a cancer that is usually misdiagnosed, and by misdiagnoses, I mean that people, when they have this form of cancer, they usually think that it's just, oh, a wart's on my skin, or oh, I got a blotch on my skin, I'm just gonna ignore it. And no, actually, these forms of cancer can be very aggressive, and if they are in a state that is incredibly advanced, it can actually cause metastasis, which is when the cancer actually proliferates and starts to contaminate all organs in the body, and it's very risky. So imagine that you have a tumor in your skin. What happens is that below the skin, we have the lymphatic system, we have the blood vessels, among many other things. But what happens is that if one single cancerous cell enters through this bloodstream, through the lymphatic system, it can easily travel to other organs and thus cause other cancers in other sites, for example, the brains, the lungs, etc. So it's very important to treat this form of cancer. It's usually treatable if the diagnosis is done rapidly before it's in an advanced stage. So that is why it's very important to keep in mind how this cancer looks, because once you see the cancer, it's go to the doctor, get yourself tested, and hope to God that they can treat it. So, the hypothesis steps. What exactly did we do in order to come to the conclusion that these selected genes actually have anything to do with skin cutaneous melanoma? So here what we did, we looked at the genetic data on the specific selected disease. We actually needed to find that this was the disease that we wanted to study. Then what we did was we located 2 to 5 genes that exhibited high alteration frequency and had incredible oncogenic potential, and by this we used the cBioPortal for Cancer Genomics database. We later identified genes involved in the manifestation of cancerous cells. So what we did was we looked up the list of genes that have been known to cause skin cutaneous melanoma. And among the genes that we selected, CDKN2A was actually one of them. Later what we did was we utilized the cBioPortal for Cancer Genomics, which is an open source platform, to help target specific gene pathways and mutation. So what steps did we actually took to prove this hypothesis? Well, let's start. We explored the selected studies, and by selected studies, I mean studies that have been conducted by different houses. In our case, it was the Cancer Genome Atlas that provided this through the cBioPortal for Cancer Genomics database. Then what we did was we identified 2 to 5 genes. In this case, it was CDKN2A, PCLO, MJAM, and LRP1B. We then queried each gene, and we later visualized and analyzed the available data so that we can come up with a conclusion for our hypotheses. This is a small video on how exactly we browsed the cBioPortal for Cancer Genomics database. What we did was we located the skin section, and then we put the skin cutaneous melanoma, more specifically the TCGA Pan-Cancer Atlas. And then what we did was that we put in the gene names, or the gene symbols. So we put CDKN2A, PCLO, LRP1B, and finally we put MGM. Now, the settings that are located on the top when it says select genomic profiles and select patient cases, we left it right as it was, and we then submitted the query. Just wait a little bit to load up the information. And here we are followed by the Uncoprint, which is actually very important when you're trying to study cancer genomics. So first we're going to start with the Uncoprint of CDKN2A, PCLO, LRP1B, and MJAM. Here we can see on the left side, we can actually see the different genes. We have CDKN2A, MJAM, LRP1B, and PCLO. Right after that, we can see the alteration frequencies. And by alteration frequencies, we mean that it isn't the same as it was in the original way. It was mutated in a sense. So we can see that the highest one that presented alteration frequency all throughout the samples and the patients were actually PCLO. And the least that presented mutations was MJAM. It's also important to note that each column is actually a patient. So, the colors. Each color is actually representative of the form of mutation. For example, blues means deep deletions, black means truncated mutations, green means missense mutations, red means amplification, and the gray areas or the gray columns actually mean no alteration. When we look at the mutation spectrum, we can actually see it's like a wave in between, or like an audio spectrum. And this is actually very interesting because it gives us the information on where exactly, or the base, the nitrogenous base, which were the ones that actually had the most mutations. And from what we can see and what we can visualize, we can actually see that a lot of cytosine and adenine mutations actually happen. So it's important to note that. And also cytosine and thymine. If we were to interpret the Uncoprint, we can actually see that CDKN2A exhibited a 45% alteration frequency, and by that I mean 162 samples presented a alteration frequency of CDKN2A in them out of 363 samples that were actually total taken. PCLO exhibited 52% alteration frequency, LRP1B presented 50% alteration frequency, and the MGM presented 44% alteration frequency. Now we're going to look at the cancer type summary of all queried genes. This is very important because now we're looking at the mutations and what mutations actually happen the most, what mutations happen the less, etc., etc. So this is what we're going to look. In the all queried genes section, we can actually see that out of all the genes together, we can see that most of them actually suffered mutation, which is a form of alteration type. And then secondly, they suffered multiple alterations as the second highest mutation type. If we look at each gene separately and where the alteration frequency happened, we can see that CDKN2A presented a form of alteration frequency that was high, categorized as the deletions. If we look at LRP1B, we can actually look that it presented a 47.93% alteration frequency known as mutation, and the MGM presented 40.5% alteration frequency of mutation type, and PCLO presented 50.69% alteration frequency. So from looking at this on a get-go, we can actually see that most of them suffered a form of mutation. Most of them suffered high levels of mutation. And the colors in the plots that we can actually see, or the graphs, we can see that green means mutation, blue means the deletions, red means amplification, and gray means multiple alterations. And just by looking at MGM, we can also see that it presented high numbers or high alteration frequency in amplification. This is actually a summary of what I just said. And now we're going to look at the mutual exclusivity. We can see that the mutual exclusivity tab tells us if it happens in mutual exclusivity or if it happens in co-occurrence. And a very, very important thing that it tells us is which one is significant. And by significant, we mean that that is significant that we can actually utilize to prove our hypotheses. So we can see that PCLO and LRP1B were actually had a tendency to co-occur together. PCLO and MGM had also a tendency to co-occur together, and LRP1B and MGM had a tendency for co-occurrence. And as we can see, they were actually all significant. Now we're going to look at the mutation charts. So what do we see here? What we see here is the protein domain. And from what we can actually see, the lollipop, actually the farther the lollipop is from the protein domain, and by the protein domain I mean this, it's the number of mutations that it has. So in this case, P114L, the amino acid, actually has a lot of mutations. Here is one of the annotations that were present in the cBioPortal for Cancer Genomics database, and P114L, which was the amino acid or the protein change, actually presented a lot of hotspots. What does hotspot mean? A place where there's actually possibility of being oncogenic. Like there's a lot of information on this, and you can see the annotation actually presenting this as possibly oncogenic. This is also another brief study that we conducted on the CDKN2A mutation chart, and we saw that W110 presented actually the possibility of being oncogenic, and the P114L, which was the one that we discussed, presented mostly missense mutations. Now, it's important to understand that P114L confers a loss of function to the CDKN2A protein, as demonstrated by the inability to suppress RB phosphorylation and loss of cell cycle control. In this case, it means that it's actually oncogenic in nature. This was all taken from the cBioPortal database, so the reference is going to be at the end. This is the mutation chart for PCLO, and as we can see, across the entire protein domain, there is a lot of mutations around it, and the color green means that most of them were actually missense mutations. From what we can see, we were right, missense mutations, and some of them were actually nonsense mutations. This mutation, on the other hand, for example, we have this case here, missense mutation causes, in the copy number alterations, a shallow deletion. This is some of the formats, and this is the protein change. This is the MJAD mutation chart, and yet again, we can actually see a lot of mutations around its protein domain. It's also important to understand that not all mutations are actually bad. Some mutations actually give a gene gain of function, others loss of function, depending on the mutation. In this case, most of the mutations that were classified here were missense mutations. Here we can actually see that most of the missense mutations occurred in the protein change known as R1097Z. And this form of mutation was missense, and most of it actually caused a copy number alteration of gain of function. And finally, we have the LRP1B mutation chart. And yet again, this is one of the most mutated ones. We can see a lot of missense mutations around its protein domains. Yet again, we have different proteins that were actually classified with a mutation type around them. Now we're going to discuss the plots for each individual gene. Okay, so the plots actually give us a messenger RNA versus copy number alteration. And from what we can actually see in the CDKN2A, we can see that there is a lot of deep deletions and shallow deletions around its region, and we can also see that the copy number alteration a diploid, but mostly we can actually see on the deep deletions. The PCLO, on the other hand, we can actually see a high number with amplification. The MJAM gene, on the other hand, presents a lot of gain and amplification on its copy number alterations, and the LRP1B, we can actually see a lot of gain and amplification. So now we're going to look at the copy number segments. And what exactly is a copy number segment? A copy number variation is when the number of copies of a particular gene varies from one individual to the next. Following the completion of the Human Genome Project, it became apparent that the genome experiences gains and losses of genetic material. The extent to which copy number variation contributes to human disease is not yet known. It has long been recognized that some cancers are associated with elevated copy numbers of particular genes. This was actually taken from the cBioPortal Cancer Genomics database. References will be at the end. So if we look at the copy number segment for MJAM, we can actually see, and before I explain what these things mean, each and every line means a sample, and the color has different meanings. For example, if it's light pink, it's low-level gains. If it's red, it's amplification, and if it's blue, it could either mean deletions, shallow deletions, or deep deletions. So, what we can see just by looking at it, we can actually see light pink and red. So most of it is low-level gains around its copy number segment. Copy number segment for CDKN2A, we can see a lot of deep deletions. For PCLO, we can actually see low-level gains. And from the LRP1B, we can actually see a mixture of deletions, low-level gains, and a little bit of amplification. Now, when it comes to the pathways, when we were looking at the pathways, we only were able to identify one available pathway, and that was the one for CDKN2A, which has known to be oncogenic in nature and has been known to cause skin cutaneous melanoma. So, what we saw and what we were able to identify from this one that was from this pathway that was given is that CDKN2A in a very altered frequency in a mutated state, in this case 44.6%, contributed on the pathway for TP53 mutation, and in this case 18.5%. And what does this mean? TP53 is actually a gene suppressor. But when in a mutated state, it actually becomes oncogenic in nature, which is very, very bad. And TP53 has been known to be incredibly oncogenic. It's actually present in most melanomas. So the conclusion of all of this, we can actually conclude that the mutated/altered versions of the mentioned genes contribute to the manifestation of skin cutaneous melanoma. We also need to divulge and to give in to the idea that we need further research on this. We need further research on each and every specific gene. We need to find different pathways. We need to put in further annotations. And why do we need this? Because if we are able to truly classify these genes with a oncogenic potential, then we can actually treat them with gene therapy. And by by diagnosing them before they actually cause the disease and before they are actually present in its oncogenic and mutated nature, we can actually further the life expectancy of a patient. And another conclusion is that we need to further integrate cBioPortal for cancer genomics in our classrooms, in our research environments, in order to truly further the understanding of cancer genomics. Cancer genomics is something that is incredibly fascinating, and it is usually ignored when it comes to undergrads that are not in genomics. So it's very important to integrate that into classrooms. These are actually the references, and thank you so much for your attention, and have a nice day.